My name is Christoph Vorreiter. I'm from the Institute of Plant Physiology at the Philipps University in Marburg. We're interested how plants respond to gravity, and we're in particular interested to understand how the single steps are when the plant receives the signal of gravity and then responds to it. And by introducing microgravity, we disrupt this process, and therefore we can identify particular components which do not react under microgravity condition, and that's how our research is about. Everybody might know that if you have a plant which grows with the root to the direction of the gravity vector, and the green leaves, they are exposed and they grow against the gravity direction. And if you tilt the plant, then the plant realizes that it is not longer in the vertical axis of the gravity vector, and it responds to it. And the green part goes up, and the root, it goes down, and the plant tries to reorientate and find the vertical position again. And this process is not investigated in detail so far. And we are interested to identify further elements, and we do that by exposing the plants to different levels of microgravity to identify by omitting or giving a gravity signal if they can respond or if they cannot. Now, these are our objects of investigation. This little plant, it's called Arabidopsis thaliana, and this is a model plant in botany, which we usually use to analyze basics of molecular plant physiology. And now what we do is that we place the plants as they grow, usually with a vertical orientation, so the roots go down and the leaves go up, looking for sunlight and photosynthesis. And then what we do during the parabolic flight in the drop tower, that we tilt the plants by 90 degrees. And the plants realize that something going wrong and that they are not in their typical position, and then they try to respond to it, and they grow with the shoots, the leaves, upwards, and then they bend the root downwards. And this requires communication, and this communication is somehow disturbed or blocked during microgravity. We are very pleased to have the opportunity to use all the three facilities which were provided by the TAM. On the one hand, just the drop tower, which gives us roughly 5 seconds of microgravity, or then using the catapult with almost 10 seconds of microgravity. And if the Gravitower in Bremen is established, we have also the opportunity to use very quickly, several times a day, 2.5 seconds of microgravity, which is a great advantage for us. And we were interested to understand if the delay, which is given by the gravity impulse, 10, 5 or 2 seconds, finds its response in the physiological reaction of the plants. And therefore it's very useful to have this all three together.